Hello, my dear students, and welcome back. I know your exams are just around the corner, so let's do a super quick and super easy revision of gene cloning. In the next few minutes, we're going to break it all down. Ready? Let's go.
Lakhs and lakhs of people in our country have diabetes, and many of them need to take insulin every single day. But have you ever stopped to think, where does all this insulin even come from? We can't just get it from animals anymore. The answer, my friends, is this amazing technology called gene cloning.
So here's our game plan for today. We'll quickly see why we need to do this, then we'll go step by step, get the DNA, use our special tools, make the clone, find the right one, and finish with a look at the famous PCR machine.
So first things first, why do we even bother cloning a gene? Well, it's simple. If we want to make a whole lot of something useful, like insulin, we need the recipe for it, right? That recipe is the gene. And we don't just need one copy of the recipe, we need millions.
But here's the problem. The human genome is massive. We're talking about 3.2 billion base pairs. Trying to find that one single gene for insulin in there, it's literally like trying to find one tiny needle in a giant haystack. It's a huge challenge. So our very first step is to somehow get the entire recipe book, the DNA, out of the cell. You can't copy a recipe if the book is locked up in the library, right? We need to get our hands on it first.
Now, this whole process might look a little complicated in this diagram, but trust me, we can boil it down to 4 really simple steps.
Here they are, 4 easy steps. First, you rupture the cell to get everything out. Then, you protect the DNA so it doesn't get damaged. Next, you separate the DNA from all the other junk like proteins. And finally, you precipitate it. Basically, you pull the pure DNA out using some chilled ethanol. Just remember, rupture, protect, separate, precipitate. Easy. Awesome. Now that we have our DNA, we need some very special tools to work with it. Let's call them our molecular toolkit. Or even better, let's call them the superheroes of genetic engineering. Okay, everyone, listen very carefully. This is a classic 3-mark question that comes up in exams all the time. What are restriction enzymes and DNA ligase? Let's make sure you know this answer by heart. It's so simple, Beta. Just think of them like this. Restriction enzymes are your molecular scissors. They cut the DNA, but not just anywhere, at very specific spots. And DNA ligase? That's your molecular glue. It's like the Fevicol of genetics. It sticks the pieces of DNA back together. Scissors to cut and glue to join. That's it. And look here. These pictures from your textbook make it crystal clear. On the left, you can see the scissors cutting the DNA and creating these sticky ends. And on the right, you see the glue, the ligase, joining the 2 pieces. It's that straightforward. Okay, let's keep going. We have our gene of interest, and we have our amazing toolkit. Now it's time for the fun part, actually building our special package, which has a fancy name, recombinant DNA. So here's how we do it. We take our molecular scissors and cut out our insulin gene. Then, and this is super important, we use the exact same scissors to cut open a delivery vehicle, which is just a small circle of DNA from a bacteria called a plasmid. Because we use the same scissors, the ends match perfectly. Then we just bring in our molecular glue, our Fevicol, and stick our gene right into the plasmid. And voila! This new combo is called recombinant DNA. But this little package can't do anything on its own. We need to deliver it into a factory that will make tons of copies for us. And our factory is a simple, hardworking bacterium, usually good old E. coli. This process of getting our DNA package inside the bacteria is called transformation. Now, there's a small problem. This transformation process, it's not very efficient. Maybe only 1 in a lakh bacteria will actually accept our package. So how on earth do you find that one special winner in a crowd of lakhs? We use a very clever trick called blue-white screening. You see, the plasmid we use has a special gene in it that normally turns the bacteria colonies blue. So here's a question for you, a real brain teaser. If we're looking for the bacteria that have our new gene, should we pick the blue colonies or the white ones? The answer might surprise you. Take a second. Why are the winners white? And here's the magic. When we inserted our insulin gene into the plasmid, we stuck it right in the middle of the blue color gene. So we broke it. This is called insertional inactivation. The bacteria that got our gene can't make the blue color anymore. So the white colonies are our winners. They are the ones with our recombinant DNA. And finally, let's touch upon something you have definitely heard of, PCR. Think of it as a magical photocopier for DNA. It can take one copy and turn it into billions.
I mean, seriously, after the pandemic, every single person in India has heard of the RT-PCR test for corona, right? Well, PCR is the technology that makes that test possible. See? This isn't just in your textbook, it's in the real world.
The whole magic of PCR happens in 3 simple steps, and it's all controlled by just changing the temperature up and down in a cycle.
So here are the 3 steps. Pay attention, this is another favorite exam question. First, denaturation. You heat it up to about 95 degrees, and the 2 DNA strands separate. Then, annealing. You cool it down to 55, and little starter bits called primers stick to the strands. Finally, extension. At 72 degrees, a special enzyme called Taq polymerase gets to work and copies the rest of the DNA. And you just repeat this cycle 25, 30 times. And the power of this is just wow. After only 30 of these little cycles, one single copy of DNA becomes over 1 billion copies. That's more than 100 crore copies. It's absolutely incredible. So what's the big takeaway from all this? Gene cloning is an unbelievably powerful tool. It lets us take one useful gene and make millions of copies, which we can then use to create life-saving medicines like insulin. I really hope this quick revision made everything clear. I know you can do it. Now go and ace this topic in your exam. Good luck!